think that.
I just pushed this.
That's,
I pushed the wrong button.
there you go.
Oh, thank you.
Don't push that button.
Which button did I push? Oh, I see. I see it. Okay, so basically what we had then was we had an indication that there was some kind of a lunar correlation to the spawning and an indication that they spawned at night, but no solid data. So what I decided to do with Jim was to go out to some well-known place called Hole in the Wall off Jupiter and put out a series of nets like this. Basically, this is just a little diagram of how we did it, but very simple. You've got the Gulf Stream current there, so the current was going downstream from the Goliath grouper. We went out there, we did that, and lo and behold, we got thousands of eggs. These are Goliath grouper eggs. They've been verified genetically by Matt Craig. And they're a little less than a millimeter in diameter. And so we knew it was possible, but it was too easy. We won the first shot, but after that we ran into a lot of problems. Problems with jellyfish, problems with, foul water, all kinds of things. Currents go in the wrong direction. Because what I wanted to do initially was to identify spawning sites by doing this. Just put downstream from the spawners and catch their eggs. It was way too complicated. So we decided to, we were catching these fish for their age structure anyway. We've caught about 700 off the Jupiter area now. So what we did was we figured out a way with a very knowledgeable captain, Mike Newman, off of Jupiter, on how to get these things. It's the only way you could catch 700 as we did. So this is a picture showing them coming up, struggling up, and being hauled onto the boat, up onto the ramp. And just to show you that it doesn't take much of a man to catch one of these things. These are two 13-year-old boys that caught most of the fish that day. And this is the size of the fish they caught. See, Goliath grouper have white muscle like a lot of other groupers and a lot of other fish. And white muscle is white because it has poor blood supply. And so they store glycogen. So the first few runs that they've got are just arm breaking. I mean, if you hang on, you're going over. But then they run out of gas. And, they got to repay that debt. They run out of glycogen. So that's what we wait for. And that takes several minutes and several runs. We also use a polyball, an orange float, and let them fight that for a while and then pull them in. But at that point, they're exhausted, and they just lie there on a stretcher, which we strap them down to that stretcher. There's the stretcher underneath, and those are the straps. And then we irrigate the gills with running seawater, and the strapping down on the stretcher keeps them from thrashing about. We collect stomach contents and a lot of other things, but the first thing we do is to vent them with this trocar and cannula, which was made for relieving bloat in cows. It works great on Goliath grouper. It's very sharp, but you put puncture the thing right behind the pectoral fin, below the midline, and leave the cannula in and pull the trocar out, and it vents very quickly. I didn't push the wrong one. I know, which is the wrong one?
It's down. Wait a minute.
Well, I mean, they're all jammed together. Oh, bad thumb. All right, I'll do it like this. Okay. All right. Then we took gonad biopsies. And this is a novel approach that we used. No, I don't know anybody else that was doing this, but these hand-operated vacuum pumps basically are made for automotive checking of vacuum tubes and canisters and so forth. And they're quite cheap. It's like $30. So you rig this thing up, and here I am pumping it, and you put a tube into the oviduct. And there's the oviduct right there. There's the anus, the oviduct, and then the urinary papilla is behind. So the oviduct's in the middle, or the sperm duct, depending upon the sex. very much more difficult getting a sample from males, but females was quite easy. We run this tube up in and move it back and forth and get our sample. And then those samples were preserved in formalin and some were put on ice for analysis of mercury, which my student Chris Malinowski is doing. So there's lots of things that Deb Murray, who did all of the analyzing of the samples, she also recorded POFs, hydrated oocytes and POFs. Recent POFs. POFs are post-ovulatory follicles, for those of you who don't know. Basically, when the egg breaks out of the nutritive follicle, it leaves a little scar, and that's the scar. So that means the fish, if the egg broke out of the follicle, that means that the fish spawned recently. And so the occurrence, the frequency of occurrence of these post-ovulatory follicles gives an indication of the frequency or the number of individuals that, let's say the proportion of individuals that spawned during those sampling periods. Hydrated oocytes give a similar thing, but they can appear in the, remember these are biopsies, so we're drawing tissue out of the lumen of the ovary, and so some of those can be dislodged before spawning, and some can be dislodged. It's not as good, but this is a very good way to tell. So plotting that out, these are the POFs. What we got was remember, we wanted to determine whether there was a lunar periodicity to spawning, and that's what this is all about. We wanted to see, in fact, if new moons, or let's say the new moon, let me put it the other way, if full moons, spawning is reduced, as the sound is reduced, or the sound energy is reduced, that are produced at night. And so, so we got it. It was highly significant difference between full moon percent occurrence of POFs and new moon percent occurrence of POFs. We only had a few samples here. This probably shouldn't even be plotted there. So we didn't have enough statistical power to show any distinction. But aside from that, it showed definitely that there was a lunar pattern to their spawning. And the same sort of thing occurred with the hydrated oocytes. So basically what we did was we figured out that, number one, they spawn at night. We caught the eggs a number of times. Number two, there was this lunar pattern that matched the acoustic pattern. We wanted also to see whether these fish were, so you got these aggregations off Palm Beach County. Where did the fish come from? Where does that buildup come from? So we joined the FACT group, which is the Florida Atlantic Cooperative Telemetry Group. And it's a great thing that FWC runs because they got hundreds of receivers up rivers, in estuaries, offshore, and wrecks. And basically you ante up with 10 receivers and you get to join their group, and they send you data and you send them their data, and everybody's happy because everybody gets, there's snook, sharks, red drum, all different species. So, but it was tremendously helpful for us. And I'd like to see the same thing on this coast. Then what we did was we knew the Zion train, which is a series of, it's a wrecks and artificial structure, all kinds of junk, but they like to spawn on that. So we knew that. And so we tagged 25 of them with acoustic tags on the Zion train, and to see, whether there was any spawning site fidelity, whether they came back to the same sites or whatever. And then we monitored, their migrations and so forth relative to the moon phases. What we found, by the way, Bob Ellis, who will be working soon at FWRI, was our student at FSU, did this work. He was a student there, and he wanted a good chunk of this project, and he did a great job. He's published on it, but he's building a much greater publication record now. But we found that these fish were coming some of them were coming all the way from Georgia. And of course, all from through here. And these are the spawning areas here, and they come down down the coast. There's not many Goliath grouper in the Florida Keys to speak of. Coral reef is not their habitat. They don't particularly, I mean, there are a few there, but nothing like up here. They like rocky reefs and they like big structures. I don't know exactly why. Maybe there's not enough prey, because there's certainly enough habitat on the outer Keys reefs. We dived along that whole area, and there was plenty of structure there that they could have used, but they weren't there. They weren't even on wrecks that were close to it, except maybe one or two every once in a while. So I think the major movement is from, in this area. Yeah, the longest distance was like about 500 kilometers, maybe a little bit longer. And then one was recently reported for somewhere up in Georgia. If we had receivers in Georgia, that would be great too. Okay, so what Bob worked on was the patterns of movement. Here's the distance moved in kilometers, and these distances are plotted by month. We could track these fish, and note also that the movement is very low in the non-spawning months, and then spawning months are August, September, and October. And these are full moons, and so what seems to trigger their migrations is the full moon, because they start moving on the full moon. In both, these are 2 years, 2011, 2012, and that's when they start to move. And then when they get there in August, they're still moving about. And they'll move about on different sites. I don't know what they're doing, but they do. So full moon seems to be the trigger for movement in July. Then in the new moon, once they get to these sites, these are new moons. Okay, this is new moon of August and September. They, remember, this is the Zion train. This is the site I tagged 25 of them. Okay, so there's a peak in the number of tagged fish on those structures on the new moons of each month. Now, the following year, 2012, you see the same kind of thing. It has nothing to do with date. It has to do with the lunar phase, because it's offset to the left here. The problem is there's a high mortality of these fish. Over 4 years, we lost 50% of their tagged fish. I can't explain it. It could be tagging, it could be, poaching, it could be a lot of different things, but it's not us. We're not doing it. So there's a problem with that. So I'm only showing 2. If we kept the population high, I could show they could continue to do that. So basically what we're seeing is the fish are spawning on dark nights, or they're looking at it the other way, they're avoiding spawning on full moon or light nights. And the most obvious explanation for that is that it's an adaptation to minimize predation by all these little egg predators that are around them. Those egg predators are 3 species of scad and at least 2 species of herring. There's a Spanish sardine, and then there's a round herring. Round herrings are most common in the Gulf, but they're covered with them. My opening picture shows the same kind of thing. That seems to be an obvious explanation, and the observation of one photographer, National Geographic photographer, Jen Hayes, the wife of David Dubélé, who's a well-known National Geo photographer, actually saw spawning. She stayed down on the bottom after sundown, and we did that too. We sat on the bottom, and Nick and Maury, and we were all sitting there waiting, and the fish never did anything, but she got a chance to see it. And anyway, so what she saw was this fish, presumably a female, with her vertical on this structure, this wreck, And then the fish, as the fish was moving vertically on the wreck, she said thousands of these fish just swarmed it to where you couldn't even see the fish anymore, the Goliath. And so it may have been that they detected ovarian fluid or something like this, I don't know, but they just came up like that. And then she went this zigzag motion across the sand, slammed into the sand a couple of times, went straight up, followed by two presumed males, and there was a presumed, I say that because there's no verification of this, sperm cloud that she saw. That's her description. She's the only person who's seen it, which means two things. One is that these egg predators probably are a serious impact on their reproductive output, especially in areas like the Gulf where there's not a strong current and the eggs just sort of hang above the spawning fish. And the other thing is that it's pretty clear that they're group spawner, not pair spawners, from her observation, but also from the fact that they've got huge testes. And fish with huge testes that are batch spawners like these are will produce large amounts of sperm. And so the competition for fertilization is at the sperm level. And that's very often indicative of a group spawner. Whereas pair spawners, like gag and red grouper and other species, have very tiny testes. And they meter out small amounts of sperm because they're spawning. The competition is at the individual level, not at the sperm level. And so they don't produce any more than they actually need to fertilize the eggs. And that's been shown with a lot of different species. Okay, now the final thing that I'd like to talk about, and again, we don't have a whole lot of data here. It's a correlation, but it's really a good correlation. Explanations for timing of spawning are usually related to some sort of a mismatch philosophy that Hort first presented in the early 1900s. That is, spawning is timed so that the larvae have the advantage of an ideal environment, food and temperature and conditions that advance survival. And the sad thing is that throughout all these descriptions of timing of spawning, people have adhered to that philosophy that it has to do something with mismatch. The trouble is a lot of people don't know about people that do that don't know about the juvenile habitat and how important it is for these fish for survival. So basically there's three stages that selection works on. The spawning adult, and that has to do with predation. Okay, if an adult is spawning at a certain time that exposes the fish to predation, then that's probably that's definitely not going to add to reproductive success. But at the other end is the biology of the juveniles. So the juveniles, if they have an advantage for settling in an environment that is variable, that is crucial to their development, but variable seasonally, then that may be the most important selective pressure on the scheduling of spawning. And that's what I think is going on with these guys, because here's a little Goliath here. They look a lot like the big ones, but they disappear in leaf litter, and that's what they submerged leaf litter, and that's what they settle in. So basically, with that 30 to 80 day larval pelagic duration, they can settle anywhere from sometime in September to sometime in early January. Now this shows the amount of leaf litter or leaf fall in the forest. This is at Rookery Bay, but there's just a good illustration. Other publications show the same thing. And also during December through April is the dry season. for South Florida mangroves. And we know that Goliath grouper prefer higher salinities, so that would be the ideal time to deposit them there, when the leaf litter is maximum and the salinity is highest. Okay, and that's just the next year. So anyway, so the hypothesis here is that spawning, the timing of spawning in this fish is related to the juvenile settlement habitat. And it seems like a pretty good guess. Gag has the same thing. This is a small gag grouper. We did work in North Florida showing the spawning or fertilization times by getting all these juveniles. So you get a single peak mode that occurs maximally in March, starts in February and March, and then settlement occurs in April and May. Now, the primary juvenile habitat for gag is seagrass. So when they settle, again, this shows when they enter that seagrass habitat, and the Big Bend is an enormous seagrass habitat, and when they leave to go to the offshore reefs, encompassed by these dashed lines. And these peaks here are the basically the above ground biomass of seagrass in these regions. This is right off our marine lab. This is Port St. Joe seagrass, but the Big Bend's pretty much similar. So I know it's just a correlation, but if settlement occurred like here and extended over to here, they wouldn't land in that primary habitat. It wouldn't be important to them. So the timing of February and March looks like it's driven by juvenile biology rather than any larval biology or by adult selection on adult spawners. Let's see if I get anything else. Okay, that's it. So these are all the wonderful people that worked on all this, and I've got everybody listed there who have helped. As you know, I'm retired, so everybody else does the work, and I give the presentation.